Okay, let's get started. So, we are entering now into Unit 3. It's kind of a clean break. So, I have started grading your exams. I didn't get done, but I was grading them until sometime last night, and I just gave up. But I should have those back to you on Monday. Aside from that, there's not really much in the way of announcements. We're going to start into Chapter 13 today. And there's one thing I wanted to announce. So, something to start thinking about. If we look at the syllabus, this is pretty much what I've got planned. So we are doing, starting today, which is the 17th, we are actually starting into the spinal control of movement. Technically, we're a day behind it. There's room in this table. We're fine. So we're going to be doing spinal control of movement. We'll do brain control of movement. I am planning to do, that kind of ends this unit, this movement unit, I guess, that is coupled then also to the vestibular system and somatosensory system that we just did this exam on. And then we'll do, I want to get into some more higher order brain topics. So I do want to do a unit on brain rhythms and sleep, which mostly is related to Chapter 19. There'll be some other stuff. But there are some other topics we need to pick another topic, basically, is the idea. And it should be something that's kind of above the level of just looking at a single motor system or some other, I don't want to just do visual system or auditory system. I want to do more whole brain type functioning. So sleep and brain rhythms in general kind of fit within that category. But there's many other chapters that are kind of in the vicinity of sleep. So what I'd like you to do is sometime, ideally it'd actually be great if you did this, let's say by mid-next week. Take a look through the table of contents in your your textbook and see if there's any chapters that kind of jump out at you that are kind of in that 16, 17, 18, 19, or beyond 19 range, and see if there's something that sort of jumps out at you. And I think the idea is that we'll try to generate sort of a short list of some topics, and then we'll vote and decide what we're going to do for that last week or so class that we have, leading up into Easter break and then following Easter break. Because I do want to have some bigger picture topics. We have a fair bit of buffer room in there, probably enough to cover one chapter well. But I'd like it to be something that we sort of choose by the class. So take a look at some point, see if there's some topics you might like. Something that's one of these later chapters in the textbook, cognition or some of these other things. Memory we could do. That's kind of a cool one. Stuff like that. Okay, there is a homework assignment associated with this chapter. My internets are down on my computer right now, so I couldn't actually check and verify what day I have them due. My guess is that I had them due, either next Friday, probably next Friday is my guess of when I had them due, but unfortunately I can't check. But is that when it is, 22nd? So that's Wednesday. We'll see how that's going. We might want to push it to the 24th, but we'll see. Okay, and then ongoing announcement, if you haven't yet, I've met with many of you already about your outlines. If you haven't met with me about outlines yet, I would like to meet with you ideally before the end of the month, because I think it's probably April 12th-ish is when the paper's due. So that gives you time to write the paper after the meeting. So by the end of the month would be good. It's not urgent, but sometime over the next couple of weeks would be good to meet. Okay. So this chapter is called Spinal Control of Movement. So this chapter and the next chapter are all about how do we actually use our nervous system to cause motor actions to occur, which oftentimes is of a great deal of interest to people in this room based on, we get a lot of physical therapy majors or occupational therapy majors, or even, any of the other health science or, pre-med type orientations. A big thing that we offer are dealing with have to do with movement problems, and so it's really important that we understand how movement works. How is it that our nervous system causes movement to occur? There is a pretty substantial part of this chapter that I'm not listing up here that does go into what happens within muscle cells in terms of interactions of actin and myosin and these cross-bridge cycling cognitive and all the other physiology courses. And I think it's getting a little far afield from neuroscience per se. But instead, what this chapter will focus on is trying to understand how it is that our nervous system, starting at the level of the spinal cord, controls the movement that our skeletal muscles are doing. So what sorts of circuits are present within the spinal cord? How do we signal down to cells? How do muscle cells respond directly to that signaling? But as far as like the actual contractual elements interacting, we're going to kind of leave that for other classes to do, because I just feel like it doesn't quite fit. But this is the plan of attack. So we're going to start with a discussion in general of the somatic motor system, which is the motor system designed or dedicated to controlling skeletal muscles. We will then talk about how we cause contraction to occur, and then how we regulate contractions at a nervous system level. So how do we regulate contraction strength and how long we contract? That includes a number of topics that are listed there. We are then also going to get into proprioception, which directly is tying into that somatosensory chapter, but I sort of said we're going to put that off to later. This is later. So we're going to talk about proprioception, how proprioception works, as well as two of the main proprioceptive organs that we use as examples. And then finally we'll get into talking about pattern generators, which will kind of harken back to some of the stuff we did with the sea slugs, but it'll help us, I think, to better understand how it is that we're able to generate repetitive motor patterns at the level of the spinal cord without direct input from the brain. So again, this is where I got ahead of myself. We're going to focus on nervous system form and function in this chapter. We're not going to focus so much on the actions of what happens entirely within the skeletal muscle cells. I really recommend taking a physiology course, either Bio 205 or Bio 406, to get that. And many of you have or will with your programs anyway. Okay, so what is the somatic motor system? The somatosensory system that we talked about were sort of our generalized senses from all the places around our whole bodies, especially focusing on skin. Somatic motor output is kind of similar in that it's sort of our generalized motor output. In this case, it's targeting all of our skeletal muscles. So that's really what it's about. So whenever you see somatic motor system, you're talking about the motor system that controls our skeletal muscles, which are distributed all over our body. That, I think, a lot of times for good reason, we tend to think about a lot of that control of skeletal muscles is originating up in the brain. And it is. And we're going to spend all of the next chapter talking about how the brain controls motor actions and all the different nuances of what can happen, brain damages and such, which is kind of fun. There's all these ablation experiments to try to really get at how different parts of the brain are involved in motor output pathways. But this chapter, I think, really recognizes that it's not just about the brain. It's the spinal cord also that does a great deal of the kind of the spinal cord directly does a lot of control of our actual skeletal motor output. So our spinal cord has many different motor programs. By motor programs, I mean that we have little collections of circuits that can be activated that perform specific motor tasks. And many of the motor tasks that are sort of like these repetitive motor tasks, so a good example of this is actually walking. Walking is something where we can turn on walking, and certainly we have the ability to, with our brain, directly control all the muscles involved in walking. But if you just look at like a basic walking circuit, our spinal cord can actually do a lot of that sort of action in terms of these sort of repeated contractions, extensions of the muscles of the limbs, one after the other, to cause the basic walking action. There's certainly also a lot of motor programs related to reflexive control and balance, and just monitoring and maintaining limb angles. So if I am walking along and I kind of lose my footing with one foot and put a little extra weight on my other foot, there are spinal reflexes that will immediately and reflexively cause increased contraction of my leg muscles to oppose that kind of lurching forward as I'm starting to lose my balance. So we have spinal programs that can execute a lot of motor actions, and then we can modify those commands based on descending output coming out of the brain. Ultimately, what we're interested in this chapter is how do we control muscle cells. So we are going to be talking a lot about muscles, and I know many of you had some physiology before in either biology classes or exercise phys or whatever it happens to be. So muscles are composed of many individual muscle fibers. Muscle fibers are sort of these giant multinucleated So if you zoom in on somebody's biceps, which is what this is doing, and look at the individual cells, here are all those individual muscle fiber cells. If we look in them, the picture doesn't really show it, but they're all multinucleated, because really what happens during development is you've got multiple cells that kind of fuse together to form these fibers. And the fibers have a very striped kind of appearance, or a striated kind of appearance. And that striation is representative of some of the different contractile elements that are present within these fibers. Anywhere there's sort of like these dark bands, these dark bands correspond to myosin proteins that are present in there, whereas the white bands correspond to places where there is no myosin and only actin. And what happens during an actual contraction is you have these actin and myosin proteins. Actin we've talked about before is a microfilament. They start to interact, and literally the myosin kind of walks along the actin. And as it does so, it pulls the entire cell shorter. I mean, the idea is that basically the myosin part encroaches on the actin part up here as those myosin molecules walk past the actin, and the entire fiber gets shorter. And as that happens, multiplied by thousands and thousands, that causes the biceps overall to start to get shorter. From a nervous system perspective, the reason all this is interesting is that these individual skeletal muscle fibers, cells, can't do anything and won't do anything unless they get direct input from the nervous system. That's different from all the other kinds of muscles. So like cardiac muscle really doesn't need nervous system input at all. It will, if you take cardiac muscle cells, at least some of the pacemaker cells, and you isolate them, and you stick them in a dish with all the nutrients and such they need, they'll go in there and they will just happily contract all the time without any nervous system input whatsoever. Other kinds of muscle, like smooth muscle that lines through your digestive tract or all your blood vessels, those smooth muscle cells have the ability to do some contraction without nervous system input. So if you stretch them, they will spontaneously contract to oppose that stretch. Skeletal muscle doesn't do that. Skeletal muscle is entirely reliant on nervous system input. And the interesting thing is that each individual skeletal muscle fiber cell is directly innervated by a synapse, or usually a collection of synapses from motor neurons. And you can kind of see that here. So here are axons that are coming down from the spinal cord. So this is axons from the central nervous system. And as you watch them, the axons will often split into multiple axon collaterals. And each individual muscle cell, so like this one, gets its own individual synapse. Really, it's a collection of 1, 2, 3, 4, 5, 6 synapses, you can see, from that individual motor neuron. This one gets its own synapse here, this one gets its own here, this one gets its own here. Every single skeletal muscle fiber has an individual synaptic connection with a motor neuron, and when that motor neuron fires and releases neurotransmitter onto the skeletal muscle fiber, that skeletal muscle fiber will then contract. So they're entirely, completely dependent on what's coming from our nervous system in terms of controlling how those individual fibers are behaving, and therefore our muscles are entirely dependent on our nervous system. So that's why we're covering it. The idea is that these, for the most part, how do I want to put this? Yeah. So the idea is that each of these motor neurons, these are motor neurons called alpha motor neurons, which are coming out of the spinal cord. We'll come back to that designation later, because there's another thing called a gamma motor neuron that has to do with the muscle spindle organ. Alpha motor neurons, each one will tend to split into multiple axons, multiple axon collaterals, which will then each target individual muscle cells. So it's not like if you've got, a million skeletal muscle fibers in a muscle organ like the biceps, you don't really need a million axons going to it. You might need 100, and then each one would split up in multiple directions and branch off to specifically innervate each individual muscle cell. But you do end up getting these situations where one motor neuron coming out of the spinal cord will have multiple axons that all split and will innervate multiple skeletal muscles, but that kind of serves as a unit, right? So you've got a motor neuron coming out of the spinal cord and sending an axon down, and that axon will then start to split, but it could innervate 5 muscle cells. It could innervate, maybe there'd be 20 different axon collaterals, each of which would hit a different muscle fiber. That whole thing functions as kind of a unit, where the motor neuron plus all the skeletal muscle fibers that particular motor neuron coming out of the spinal cord directly innervates. That's kind of a unit. We do call it that. We call it a motor unit. So that kind of functions as a one thing where if this motor neuron gets excited and sends a signal down its axon, which then goes off in all the different axon collaterals, all the skeletal muscles that it's attached to will all simultaneously contract in response to that motor neuron getting activated. So collectively, it sort of functions as one big unit. If you, and then there's another term that's related to that. So if you're looking at one individual muscle organ, again, I'm just pointing at the biceps because that's the example that's shown here. That one muscle organ will have a collection of motor units, each of which corresponds to one motor neuron and all the skeletal muscle fibers it innervates. That whole collection of motor neurons, or that collection of motor units, might be another way to say it, that corresponds to a motor neuron pool. So in order to activate my bicep, I can basically make a decision, or my nervous system can make a decision. Am I going to activate 5 of the motor units within that motor neuron pool? Or am I going to activate all of them within the motor neuron pool? If I only activate 5, it'll result in a pretty weak force of contraction that won't cause rapid movement or won't generate a lot of force. If I activate everybody, all the motor units that are present within your motor neuron pool, that would cause every single muscle fiber in the muscle to contract simultaneously, and you get a much harder, aggressive, forceful kind of an action. And we have the ability to modulate. And that's one of the things we'll be talking about, is how that modulation occurs. Okay, so that's kind of like that's big picture overview of somatic motor system. And what we're going to do then is try to sort of walk through, starting at the spinal cord, walk through some of the components of that system. Okay, so we're going to start up with the spinal cord, as I mentioned. Usually what will happen is if you're trying to execute some sort of motor action, there is a plan for a motor action that's going to originate up in the brain. And that's kind of the next chapter to understand how that happens. But the brain then, from some areas of your frontal lobe, will activate axons that will travel all the way down your spinal cord to a particular level of your spinal cord. And those first motor neurons coming out of the brain, going down your spinal cord, and ultimately, that are carrying information about a motor plan, we call those things upper motor neurons. So those are traveling from the brain to the spinal cord. They would travel down ventral or lateral tracts, and we'll talk about some of the specific tracts that these things travel within. But they'll then the axons will enter into the gray matter, and they'll go into the ventral horn of the gray matter, which is where all the lower motor neurons are found. So the idea is that your upper motor neurons send impulses down to the lower motor neurons. They innervate these and form synaptic connections with these, or at least with interneurons nearby. That ultimately then causes the lower motor neurons, which are housed here in the ventral horn of the gray matter, to get stimulated. And as we saw back in chapter 7, ventral horn of gray matter that is entirely composed of these motor neurons. So this is where we have these motor neurons leading to skeletal muscle housed. They will then give off axons, which will carry information out the ventral root of the spinal nerve, through the spinal nerve, and then ultimately those axons will travel to muscle organs, where they'll start to split off into collaterals, and ultimately you'll have innervation of individual muscle fibers down here. So it's the lower motor neurons that emerge from the ventral horn of the spinal cord that are directly forming synapses with the muscle fibers. There are 2 classes of these kinds of motor neurons. So of the lower motor neurons, there's 2 varieties. The main ones are the alpha motor neurons, which that term was on the prior slide as well. Most muscle fibers are innervated by alpha motor neurons. These are the ones that are causing, because they innervate most of the muscle fibers, these are the ones that cause the majority of the force of contraction, the vast majority, like 99.8% or something, of the force of contraction when you're actually contracting your muscles. And that's due to alpha motor neurons stimulating muscle fibers, causing them to get excited and then contract. We also have something called a gamma motor neuron, which we're going to see on the homework primarily. The gamma motor neurons are responsible for innervating muscle spindle organs, which are a proprioceptive organ that's found within the muscle itself. These things are basically stretch or length sensors, is often how they're described. So what they'll do is that as a muscle as you place additional load on a muscle, you'll start to stretch the muscle. You start to basically cause the muscle to increase in length. And as a result of when that occurs, when the muscle is starting to increase in length due to load being placed on it, that will cause these muscle spindle organs to start firing. But there's sort of a problem in that muscles are not kind of one set length, right? So, if we just look at my bicep again, in this state my bicep is quite long, but if I contract my bicep and allow my arm to flex, at that point now, this muscle has shortened in length. And it's shortened by, a couple centimeters probably over the length that it had been before. So what these gamma motor neurons do is that they stimulate little tiny muscle cells that are housed within the muscle spindle organ that cause the muscle spindle organ to change length in a way that corresponds to the overall length of the muscle itself. So you can think of them as like embedded sensors that will change their length to calibrate their length to the overall length of the muscle. We will get back to that point in homework, so I'm kind of setting it up for now, but there's a good figure that a reference on the homework that you can work through. Mostly we're going to be focusing, though, on the alpha motor neurons in this chapter. So, if you're looking at an alpha motor neuron and you're trying to understand what kinds of signals is it receiving, where is it getting its different signals from, there's actually a sort of a set of them. So the main one that I tend to think about is, so again, we're looking at alpha motor neuron. This is carrying signals down to skeletal muscle fibers, and when stimulated, this alpha motor neuron will cause the muscle fibers that it's attached to contract. So we're getting muscle movement as a result. These guys do get lots of input from the upper motor neuron. So I tend to think of like the main pathway for typical movements is you get a signal up in the brain going down the upper motor neuron that causes the alpha motor neuron, which is a kind of lower motor neuron, to carry its signal to the muscle cell. But you'll also have other kinds of inputs that are coming in. So for example, we'll get input from the muscle spindle proprioceptors themselves. Again, this is kind of getting ahead of ourselves, but all this stuff is connected. But the idea is that when you have a muscle that's starting to get stretched, your muscle spindle organs can detect that change in length, and as a result, they'll send signals up which can actually reflexively cause these alpha motor neurons to increase their signaling and cause the muscle to kind of contract to oppose that stretch. So if you're like trying to force your arm to stay at this angle, even though some jokester is coming up and like grabbing your arm and pulling it down, you have a reflexive circuit that's set up where these muscle spindle organs will detect that your muscle is starting to increase in length, send signals back up, which will innervate the alpha motor neurons and cause you to contract even harder to oppose that increase in length. So that's one thing, sensory input from muscle spindles in addition to input from the upper motor neurons from the brain. You'll also have input from spinal interneurons. And these spinal interneurons can be conveying other kinds of input. So there are other reflexes that are associated with, that are operating through your spinal cord beyond just that muscle spindle one. So those kinds of reflexes will be feeding into and informing alpha motor neurons about what to do. There's also, there are little motor programs, like we talked about, little walking programs, for example, that are present within the spinal cord, that also can be feeding into the alpha motor neurons and causing them to contract at certain times. So it's not just sort of a brain to spinal cord to muscle sequence, even though I tend to think of that as like the main sequence, maybe the easiest sequence to understand. Is that okay so far? Questions on anything? Okay. So here we're showing like one segment of the spinal cord. As we talked about in the somatosensory chapter, chapter 12, we don't just have one sort of segment of the spinal cord. There's a series of these that occur. As you move down the spinal cord, you go through segment after segment of the spinal cord. So there's 30 spinal nerves in all, each of which corresponds to sort of an individual segment. So you can think of the spinal cord as being divided kind of into 30 different segments. And each of those does have a ventral horn of gray matter that is the location of cell bodies that will carry motor information out in that sort of general region. So, my cervical spinal nerves are carrying motor output that's going to muscles that are kind of in the region around my neck, whereas my lumbar area is carrying spinal nerves that are kind of in the region that's going to go out into muscles around my torso or down into my thighs, and so on. if you do look at cross-sections through the spinal cord, I always thought this was pretty cool. You do see that the spinal cord does look different in cross-section depending on where you are as you move down the spinal cord. So the idea is, in fact, even just looking at it from behind, it changes in width. So as you're moving, we start up here, very much right at the base of the brain, and then start working down. We go through a pretty narrow region through much of the initial sort of layers of the cervical vertebrae. But then we do go through this thing called the cervical enlargement, which spans from spinal nerve C3 all the way down to spinal nerve T1. And you can see it's just visibly thicker in that area. Then we go into another area that tapers off and becomes a little bit more narrow throughout most of the thoracic area, and until we get to the lumbar area, where we see another enlargement. It's called the lumbar enlargement, where we get a thicker spinal cord in that area as well. And the main thing that seems to be happening here, maybe you can sort of think about it. Like, what do you think this part of the spinal cord could be controlling, and this part could be controlling in the cervical and lumbar regions that wouldn't be being controlled in this thoracic region? What could be different in terms of, if you just think in terms of skeletal muscle, what could be different in those two areas that would cause this area to need to be thicker and have more neurons?
Got the arms and legs there.
Right, yeah. I think this is the big thing, is that we've got up here, if you do a cross-section, what you end up seeing is that there is a pretty large dorsal horn. But the big thing is you've got a very large ventral horn of gray matter within your cervical enlargement. Ventral horn of gray matter is the location where you have motor neuron cell bodies that are carrying information to skeletal muscles. Right? If you go further down into the thoracic region and take a cross-section, what you end up seeing is that the ventral horn is much smaller. It's like this piddly little thing, right? This is still the region where you've got control of skeletal muscles, but what this would probably tell you is there are fewer skeletal muscles that this area is controlling than up here. And the reason is that, up here, this is the location of cell bodies in all these segments, C1 through C3 through T1. This is where you have cell bodies that are sending information not just to the skeletal muscles of your torso up in this region, but also all the way to all your various arm and hand muscles. I mean, there's many, many muscles out there, tremendous number of motor units, and each of those motor units has an individual cell body that's housed within the ventral horn within this sort of narrow region of cervical enlargement. As you get in the thoracic area, really the only skeletal muscles these areas are controlling are kind of your abdominal torso muscles, right? So you do have muscles there. You've got your spine, you've got your obliques. So there are muscles to control, but there's far fewer of them and far fewer motor units. So there's fewer cell bodies needed, and they just don't take up as much space. So the spinal cord starts to look narrower. You also notice there's not as much of dorsal horn or gray matter. Dorsal horn is where all the sensory input's coming in. So you also have less sensory input coming in because you don't have tons of really fine touch receptors and such in your torso. Whereas, up in the cervical enlargement is where all, you are getting, in the dorsal area, you are getting some input coming in from your touch receptors of your fingers, at least forming some small reflexive sorts of circuits that are present in the spinal cord. Once you get down to the lumbar enlargement, here's where you start to have control over your leg muscles. So again, you have a nice enlargement of your ventral horn. So it's a pretty visible change in the spinal cord. The spinal cord is not homogeneous as you're moving down, and it seems to correspond in large part to how many muscles need to be controlled in each of these different areas. Now, one thing that your book doesn't really show is that once your axons emerge from the ventral horn of gray matter, they go out through the ventral root, they go out through the spinal nerve, but they don't, once they form the spinal nerve, they don't kind of make a beeline to the targeted muscle organ. Instead, they undergo these sort of really complicated branching patterns called plexuses, and that's what I'm trying to show here. In fact, it kind of says this here. So oftentimes, I'll go back to that in a second, sorry. Once spinal nerves emerge from the spinal cord, so the spinal nerves, remember, that's the mixed thing that actually passes through the intervertebral foramen, they will then fuse to form a plexus. And basically in plexuses, you have this sort of fusing and then branching and the fusing and branching of nerves coming out of the spinal cord. So in this picture, this is showing the brachial plexus. So on a person, the brachial plexus is basically this set of spinal nerves. So the spinal nerve would be like, right where I'm trying to hold my cursor. That little tiny spot, that's where the spinal nerve is. And immediately, once you're out of the vertebral column, you start to have this thing that occurs where these fibers start to fuse, and then they branch, and fuse, and they branch, and they do so in pretty complicated fashion. So in this case, you can see that here's spinal nerve C5 coming out. It immediately starts sending off these branches where axons are kind of going off in different directions. And a big chunk of them come down, and they fuse with a set of axons that came from C6, which then in turn branch several other places, and some of them will end up going out the musculocutaneous nerve, some will end up going down the median nerve, some will end up going out these nerves that aren't even labeled here. It gets to be very complicated, and I think some of you have to study this ad nauseum. I don't care that you know the details of like the branching for this class. That doesn't matter. But I do want you to understand that there is a branching thing that occurs, that you get these complicated plexuses. And you get this occurring up here where you have your cervical enlargement. So all these spinal nerves here, these are all spinal nerves that are emerging out of that cervical enlargement that ultimately are forming the nerves out here that travel down your arm. And then, like each of these nerves contains a bundle of axons, a large bundle of axons, that will directly then innervate skeletal muscles that are present within your arm. And different ones of these nerves tend to go to different areas. That doesn't sound very good. Grammar wasn't very good enough. You get the idea. Each nerve goes to different collections of muscles and therefore powers different parts of your arm. You don't have plexuses occurring in the thoracic region. So it pretty much, you just kind of get these one-to-one spinal nerves go off, and then they go their separate ways directly to the muscles they need to innervate. But then you get another plexus once you hit the lumbar region, and that's most of those are going to end up going into the leg. And similarly, you have a sacral plexus as well. So again, I don't need you to reconstruct this. I just want you to be aware that plexuses exist, what they basically are. And the result is that, you've got axons coming from, if we could just sort of list out as one example, going into the musculocutaneous nerve, you have axons from C5, C6, and C7 that are all feeding ultimately into the musculocutaneous nerve here. So it's not just one level that's sending axons out to a particular skeletal muscle. Okay. So there's some organization in terms of the routes of spinal nerves. There's also some interesting organization that occurs if you look within the ventral horn of gray matter within any one segment as well. So in this case, what this figure is trying to show is that, so here we're zooming in on one part of the ventral horn of gray matter. And it's not, the point of all this is just like we saw within this metasensory system and the mapping that exists within the brain, it's not just sort of a random distribution in terms of where particular cell bodies are that are going to power particular kinds of muscles. Different parts of this ventral horn will tend to go to different sorts of muscles. So this would be a cross-section of the ventral horn of gray matter somewhere within that cervical enlargement because we're going to be sending information out to the arm. And if you just start going through and mapping and saying, all right, so what are these kinds of, the cell bodies of motor neurons that are found here, what are they going to, what kinds of muscles are they going to, versus the ones that are down here? You see some interesting patterns. So for one thing you often encounter is that most of the motor neurons that lead to extensor muscles, as well as some of your axial muscles, so extensor muscles particularly related to limb extension. So extension is this kind of action, or it can be this kind of action. It sort of depends on the motion that we're dealing with. But most of those flexor muscles tend to be found more, I guess I could say, so more in the dorsal part of the ventral horn, or the more proximal part, the part closer to the center of the gray matter of the spinal cord. Whereas if you want to find extensor muscles that tend to be located a bit more distally, you tend to find those more in these areas down towards the tips of the horn. So the more ventral part of the ventral horn tend to have a lot of distal muscles and extensor muscles. And there's a bit of a lateral, axial, medial versus lateral thing. So axial muscles tend to be also found a bit more medially. Distal tends to be found a bit more laterally. There's some overlap here. So distal muscles tend to be a bit more loaded towards extensors. Flexors tend to be found a bit more axially in general. So there's kind of a gradual gradient as you're moving from this part of the gray matter down into this one. The main point of this is that there's sort of a mapping here. It's not randomly distributed. You could sort of map across your arm, and you end up finding that down here you've got mostly your distal and your extensor muscles, and then as you move further up your arm, get more into axial muscles, you're also moving up into gray matter in terms of where the cell bodies are that control those muscles. Any questions? All that stuff. So that kind of gives you an overview, I guess, largely of the anatomy of what we're going to be dealing with here. So how the somatic motor system is set up, what the relationship is between the skeletal muscles and those spinal nerves, and how the spinal nerves travel from the spinal cord down into skeletal muscles. What we want to do now is transition into talking about how we actually activate those muscles, and ultimately then also how we regulate contraction strength and contraction time. So I think to kind of set this up, there's sort of an assumption that I'm sort of assuming that you guys understand, and that is that motor neurons interacting with skeletal muscles is really very similar to any kind of a neuron interacting with its target cell. In that, you have your motor neuron gets excited, sends an action potential all the way down its axon. That then gets, that action potential reaches the synaptic terminal, which is the connection between the motor neuron and the skeletal muscle, and at that point, then you have neurotransmitter being released. We did talk about acetylcholine being released, for example, at these neuromuscular junctions, which in turn is going to cause a response in the muscle cell and cause it to start contracting. Okay, so it's, but the point of this is it's still, action potential down an axon that causes release of neurotransmitter that causes then a response in the targeted cell. The question, once we understand that, is how is it that motor neurons can vary how they're stimulating the muscle cells themselves, such that we can get different kinds of contractions. So for example, if you just stimulated this motor neuron and sent a signal to that, you can sort of imagine, well, I'll just explain to you. This is a little, a common type of preparation where a lot of times it's done with like a frog gastrocnemius muscle. It's a really nice preparation for this. But the idea is that you, in this case, they've basically taken a skeletal muscle and they can isolate it between a prep. They can measure the tension that's placed on this force transducer as you have this muscle that's sort of suspended between these two metal plates. So when this muscle contracts, you can detect with a force transducer down here how much force is being placed on the platform as a result of the contraction. And then the nice thing about that preparation, it's pretty easy to isolate the nerve that's leading into that particular muscle, so you can then apply currents into that nerve and cause a muscle contraction. If you just apply one little burst of current, so you kind of stimulate the motor neurons one time, what ends up happening is you get a little twitch, and that's what they're showing in this picture here. So signal is sent down the axon, the motor neuron then releases neurotransmitter onto the skeletal muscle cells, and then the muscle cell contracts. And as it does so, you get this little burst of activity, this burst of force that's being generated by this muscle cell, and you're picking it up and measuring it on your force transducer. So that's what you're detecting here. It's this little surge in force that corresponds to one electrical stimulation of your presynaptic neuron. Here, then they do it again. After the thing is returned back to its resting amount of force, so it's not applying any force anymore, you stimulate it again and you get another little blurb of force. That little blurb, whatever that is known as a twitch. So that one stimulation causes a twitch. And twitches tend to be fairly characteristic, as long as you don't overdo it. A given skeletal muscle will generate just a relatively small amount of force, very uniform, but it's a uniform amount of force each time you cause it to twitch. And it's a pretty uniform length, too. It doesn't last for very long. The thing is, our muscle cells don't typically operate in twitches, right? I mean, occasionally I'll get something like a twitch, like when I'm tired, my stupid eyelid muscles start to twitch and fidget on me, that kind of thing. But for the most part, we don't have very twitchy kinds of actions. Instead, mostly, we create nice, smooth, continuous types of actions. And so what this means is that this kind of preparation alone, where we're individually sending individual impulses down to the skeletal muscle here, that cannot account by itself for the kinds of contractions we usually get. Instead, we tend to have nice, long, continuous, graded contractions. So that's what this is asking about. How do we get nice, long, continuous, graded contractions? So graded contraction means it can occur over long, extended periods. It also means it can occur over variable amounts of force, and it's not herky-jerky. It's these sort of nice, continuous kinds of motions. So how do we do that is sort of the next this question. Let me just read through this because I feel like I approach this differently. So I think mostly we said all this stuff, but the idea is normally if we just stimulate one time, alpha motor neurons would release acetylcholine out of the skeletal muscle. You release a massive quantity of acetylcholine, so it's not like in the central nervous system where you release like 1 vesicle worth of acetylcholine. You're just dumping 200 vesicles onto the neuromuscular junction. And as a result, you get a massive action potential occurring within the skeletal muscle fiber. That action potential is always sufficient in order to send well, really it's a barrage of action potentials that reverberate up and down the skeletal muscle fiber. That in turn leads to calcium release, which causes contraction within the skeletal muscle. So how are we going to regulate that? One of the ways I think that probably is immediately apparent as you start looking at this is you could say, well, if I wanted to not just have individual twitches, I could stimulate before we completely relax. You know, here we're waiting until we're totally relaxed before we stimulate again. So what happens if I stimulate more frequently? That's what's being shown here. So again, this is the same picture from the last slide. That's an individual twitch. So one stimulation leads to a characteristic small amount of force generated, and then the muscle will relax in a pretty characteristic period of time. But if you start to stimulate more frequently, so you stimulate before the muscle cell completely relaxes, then what ends up happening is a kind of temporal summation. It's not exactly the same type of temporal summation we were seeing before, because we saw temporal summation when we were talking about synapses, more dealing with like gradual building up of EPSPs. Here what we're doing though is it's kind of the same concept. We're stacking twitches kind of on top of each other by stimulating them, the muscle before it can relax all the way again. So we kind of build gradually this sort of sequential amount of force that we're generating. And so in this way, now we're not locked into only being able to cause, whatever this would be, something like maybe half of a second length of a contraction. Now we're doing it, we're able to basically contract indefinitely. We can have long-term contractions that are occurring over a period of time by just stimulating before the muscle ever gets a chance to relax again. We call it temporal summation. And this is kind of a low-level temporal summation, but it can get to be much higher. So in this case, what you're seeing here is that we've increased frequency even more in terms of how often we're stimulating the motor neuron. And here we're stacking on top of each other such that we're almost not giving it a chance to relax at all in between stimulations, right? So this is sort of first twitch, second twitch, but then just as we start to relax from the first twitch, we have a second twitch occurring. And we end up getting, if you increase temporal summation or increase your frequency of stimulation high enough, you kind of get to a peak amount of force that the muscle is tending to contract. So at this point, we call these two things both are sometimes also referred to as incomplete tetanus. That's to contrast with true tetanus, which we don't tend to usually see, which is where if we even increase the frequency of stimulation even higher, then we get a situation where there's literally no opportunity to relax at all in between stimulations by the motor neuron. And so you get this huge amount of contractile force. You're basically contracting this muscle cell maximum with absolutely no wavering, no unevenness at all, because you're basically telling it to contract at the highest amount of force you can ever contract at any one time. And that's at an individual, all these are individual muscle cell contraction levels that we're seeing. Now, this never happens in real life, unless you've got some kind of pathological condition. So tetanus, where else have you guys heard about tetanus? Have you ever gotten one? Vaccination? Called tetanus, right? So tetanus vaccinations, you're not trying to vaccinate against this ever happening per se. What you're vaccinated against is a bacterium. Clostridium, is that one? probably, I think it's clostridium. It releases a toxin that causes lockjaw, and basically the disease is called tetanus. So when you're taking a tetanus vaccine, you're trying to prevent exposure of yourself to that bacterium that releases a toxin that basically causes this to happen. So the condition tetanus is sometimes called lockjaw, because literally what's happening is some of the muscles of your mouth are seizing up because they're so excessively stimulated, your muscles are basically in tetanus. That won't happen in non-pathological conditions, though. Usually we tend to operate our muscles somewhere in this range. And so in order to have a nice continuous contraction, if I'm doing something really nice and graceful, which most of the time we're doing something relatively graceful, what will be happening there is that our individual muscle cells, if you track them, they're doing this kind of thing, where they're kind of cycling somewhere between a twitch and true absolute tetanus. They're kind of like doing this, or they're doing this. So they're having sort of a continuous long-term period of contraction. We don't notice the up and downs, right? So this would make it look like it would be sort of a really jerky kind of a contraction. We don't tend to notice that because this is one individual muscle fiber. And other individual muscle fibers might be doing the same thing nearby, but they're going to be a little out of sequence. So while this one is relaxing, others nearby might be contracting. And so that all kind of evens out at the level of the whole muscle organ itself. But they are able to contract for extended periods, and that's because we achieve this incomplete tetanus / temporal summation. Any questions on that? It's a good stopping plot. This next figure, I don't know, that always takes me a second to get through. So we're going to stop there. We'll pick up on Monday. I actually think Wednesday having homework due is going to be good. That seems like pretty good timing based on where we are. So it's not as big of a chapter as I was thinking. So we'll still plan to do homework on Wednesday at this point.